Hello everyone, welcome back to another learning lesson. Today we are going to simplify enzyme and its classification. By the end of this video, you will have a clear conceptual understanding that's useful for your exam as well as the clinical applications. Before we begin, if you find this video helpful, please like, share and subscribe to support this channel. Now let's understand enzymology step by step. First of all, we define enzyme. Enzyme may be defined as a biocatalyst synthesized by the living cells or living organisms. They are protein in nature, exception is RNA, that are acting as a ribosome, and they also colloidal in nature, thermolabile in character, and specific in their action. And this is an example where why enzyme is important. In the laboratory, hydrolysis of protein by a strong acid at 100 degrees Celsius takes at least a couple of days. The same protein, if you have to fully digest by the enzyme in GI tract, gastrointestinal tract at body temperature 37 degrees Celsius, it takes only couple of hours. So this is remarkable difference. In clinical reactions taking place in the living system is exclusively due to this enzyme. Now, historical prospects. If you see Berzelius 1836 coined the term catalysis. Catalysis means dissolve, and now then 1878 who has coined the term enzyme, and Wagner also 1883 he was died and achieves the isolation of enzymes. Now, James Summer first achieved the isolation and crystallizations of the enzyme from urease Jagbin and identification in 1926. So this is the historical prospects how enzyme was discovered. Now to see nomenclature and classification. Actually, earlier days the enzymes were given names by the discoveries arbitrarily manner. For example, trypsin, chymotrypsin, they convey no information about the function of the enzyme or nature of the substrate on which they act. Sometimes the suffix AAC was added for substrate for naming the enzyme, like lipase, like nuclease, lactase, etc. So these are known as the trivial names of the enzymes. But now uh, there are one international organizations they can explain the nomenclatures. Enzymes are sometimes considered two broad categories. Number one, intracellular enzymes, they functions within the cell and where they synthesized. And another is extracellular enzymes. These enzymes achieve outside cells and all digested enzymes belong to this group. Now, IUBMB or IUB, International Union of Biochemistry, appointed an enzyme commission in 1961. This committee made a thorough study for the existing enzymes and devised some basic principles for the classification and nomenclature of enzyme 1964. And enzymes classified on that basis, there are six types, six major classes. What are these classes? First class is called the oxidoreductase. Enzymes involved in oxidation reductions. From the name we understand oxidoreductase. And transferase, that means transfer catalyze the transfer of functional groups from one enzyme to from enzymes to another part. Hydrolases, enzymes that bring about the hydrolysis of various compounds. Lyase, that means enzymes specialized in addition or removal of water, ammonia, carbon dioxide, etc. Isomerase, isomerization reactions, it helps. Ligase, it means it helps to ligate it or joined together with the help of ATP. Now, this enzyme commission has proposed some things, that is first class, first digit, then second class, then subclass, that is second digit, subclass, that is third digit, and individual enzyme fourth digit. In this way, they can name the enzyme. Like this, actually, EC 1.1.1.1, that means one is the class, then another one is called the subclass, then another one is called the one is the subclass. As per IUBMB classifications, what you see, first digit is called representing the first class, second digit stands for the subclass, third digit represents the subclass or subgroup, and fourth digits gives the number of the particular enzyme. Like oxidoreductase, the group of enzymes will catalyze the oxidation in one substrate with the simultaneously they reduce the another substrates. So oxidoreductase, these are the example: lactate dehydrogenase, glucose 6-phosphate dehydrogenase, succinate dehydrogenase. And second enzyme is called the transferase. Transferase, that means this class of enzyme transfer one group from the substrate to the another group. This is a reaction which we want to understand, that hexose plus ATP is hexose 6-phosphate plus ADP. The enzyme is called the hexokinase. Number 3, hydrolases. This class of enzyme can hydrolyze ester, either peptide or glycosidic bonds by adding water and then breaking the bonds. This is the example: acetylcholinesterase. Now, number 4 class is the lyase. That is, the enzyme can remove group from substrate and break bonds by mechanisms other than the hydrolysis. That is called fructose 1,6-bisphosphate, glycerylate 3-phosphate, dehydroxysteroid phosphates. So this is the total number of six classes of enzymes starting from oxidoreductase to ligase. Now, another important thing, in that way, synthase and synthetase, what are the difference in between these things? Actually, synthases are the ATP-dependent enzyme catalyzing biosynthetic reaction. They belong to the classes of ligase, number 6 classes. Example as carbamyl phosphate synthase, or argininosuccinate synthetase, or synthetase glutamine synthetase. That is all of the example of synthetase. That means ATP-dependent enzyme is called that is called synthetase. But what is the meaning of synthase? Synthases are the enzyme catalyzing biosynthetic reaction, but they do not require ATP directly. They belong to the classes other than ligases. Example as glycogen synthase, alanine, or delta aminolevulinic acid synthase. All of the example of these things. So this is enzyme classification and enzyme definition. And one important thing we want to highlight here: characteristics of the enzymes. Now, enzymes, almost all the enzymes are protein in nature, and enzymes follow the physical as well as chemical reaction of the proteins. They are heat labile, they are also water soluble, and they can be precipitated by the protein precipitating reagent, and they contain 60% weight of nitrogen. So this is the main important features or characteristics of an enzyme. Now, there is another important thing is called the coenzyme. What is coenzyme? The enzymes may be simple protein, complex enzymes, and containing non-protein part, that is called the prosthetic group. Actually, prosthetic group is called the coenzyme. It is heat stable, and the protein part. If you see a protein, an enzyme, there is two part. One is the protein part, another is the non-proteinous part. Non-proteinous part, which is called the prosthetic group, that is called the cofactor or coenzyme, and another part, protein part, is called the apoenzyme, that is protein. It is heat labile. And if you combine these two things, that means prosthetic group and apoenzyme, then it is called the whole enzyme, that is called whole enzyme. So these are the salient features of a coenzyme. The protein part of the enzyme gives the necessary three-dimensional infrastructure for the chemical reactions, but the group is transferred from or accepted by the coenzyme. And the coenzyme is essential for the biological activity of the enzyme. Coenzyme is a low molecular weight organic substances, and it is also heat stable. So these are the important features of a coenzyme. So in this video, we want to highlight here, we already highlighted that the definition of enzyme, moreover the classification of enzyme, some historical prospects of enzyme, and also coenzyme, definition of coenzyme, what is prosthetic group, what we may call the hollow enzyme, and everything has clear, I think, in this video. So we'll So thank you for this watching of this video.